called DESTINY-Breast06. Can you tell us a little bit about that design and top-line data?
Yes, so DESTINY-Breast06, as you mentioned, enrolled patients with not HER2-amplified disease, but low expression of HER2. So this was essentially defined as a IHC of 1+ or 2+, and not amplified by FISH. The trial also enrolled patients with a new terminology, ultra-low. And HER2-ultralow, of course, are those patients who may have been previously classified as IHC 0,
right.
But you see interspersed membranous staining, essentially. And what was interesting about the trial, it also sort of moved up T-Dxd a little in terms of lines of prior therapy. So really addressing the question, should we be thinking about using T-Dxd right after endocrine therapy resistance?
I see.
And then can we also include our ultra-low patients?
So as a first foray into kind of cytotoxic,
Yes.
instead of chemo,
Yes.
because that was a control arm.
That's right.
Chemo du jour,
that's
so to
right.
speak.
This was a randomized phase 3 trial with the standard of care arm being chemotherapy.
And it's
of
a positive
physicians' drugs.
study.
It is a positive study, so there was a progression-free survival benefit using T-Dxd in that population versus physician's choice chemotherapy.
And the ultra-low subset analysis kind of mirrored the intent to treat population as well.
Yeah.
Even though it was a smaller subset analysis, so not robust statistics necessarily for that group, but
Right.
the trend seemed to be the same. And as a result, the FDA has label indications for both low and ultra-low.
That's right. I mean, I think a strength of the study design, as you mentioned, it was the ultra-low subset was a smaller sample size or population, but this was a pre-planned analysis.
Exactly.
And so I think that helps to bolster the results a little bit.
Interesting.